Hello guys, welcome back to my channel. Hope you all have subscribed my channel. Now let's go. We will study today an amazing topic called photosynthesis. So photosynthesis is a process by which plants makes foods with the help of chlorophyll, which is produced by chloroplast, and these foods are stored in the granules form in the areas of the plant cell. So let's go. We'll see the mechanism of the photosynthesis. So photosynthesis occur in the tiny structure inside the cell called chloroplast. Chloroplast is the area where the photosynthesis process occurs. The chloroplast carry out the light reaction in the thylakoids and dark reaction in the stroma. In chloroplast there are the two structures, thylakoid and stroma. In stroma the dark reaction take place in which the light is not a necessary, and in thylakoids occurs a light reaction in which the light is compulsory reactant. So photosynthesis involves the light dependent reaction having non-cyclic and cyclic photophosphorylation and dark reaction where energy is utilized. So these are the process by which the photosynthesis process occurs. So is the source of oxygen. By looking at the simplified equation of the photosynthesis, it is not clear whether the oxygen produced comes from carbon dioxide or water. For a long time, it was assumed that the oxygen produced during photosynthesis came from carbon dioxide and the carbon combined with the water to form the sugars. As we know, the sugar, the major part of glucose is formed in the photosynthesis process from 6CO2 plus 6H2O in the presence of light and chlorophyll, which is the pigment which is present in the chloroplast. We get the glucose formed and oxygen as a byproduct which we inhale. There were various experiments which were carried out to show the photosynthesis process, to prove the photosynthesis process which occurs in the leaves. So there were mainly three experiments, three person who carried out the experiments. First was Van Niel, then we have Ruben and Kemen, and at the last we have Robert Hale. So we'll understand about how they conducted the experiments in a short form. First experiment is experiment by Van Niel. In 1931, Van Niel showed that the photosynthetic bacteria do not produce oxygen though they use the carbon dioxide. So he gave a hypothesis that all photosynthetic organisms need a source of hydrogen. So if we see in purple and green sulfur bacteria, he showed that the sulfur was released. From sulfur bacteria, the sulfur was released but not the oxygen. So meaning that the CO2 was not the split. So rather H2S was broken down and the hydrogen reduced the CO2. So here we get the equation that the other equation, in the presence of water we get this equation, in the presence of H2S we got this equation. In this also we get the oxygen but in the water form. So this is the 6CO2 plus 12H2S. In the presence of light and chlorophyll we get glucose and 12 atoms of sulfur and then 6 atoms of water. So based on the same principle, it was assumed that water gets split into the hydrogen and oxygen released as the byproduct, while hydrogen combines with the CO2 to produce sugars, mainly carbohydrates. So it also provides that water is produced as well as using the photosynthesis. So water is also produced with the help of photosynthesis process. Then we have the experiment by the Ruben and the camel. In 1941, by using the Van Hel's hypothesis, the rare isotope of oxygen, O18, oxygen of with having 18 atomic mass was used. Scientists Samuel Ruben and Martin Kemen conducted the experiment and provided that all oxygen evolved comes from the water only. So this all were which the oxygens which were evolved, this all comes from the water molecule which gets split into the hydrogen and oxygen, and hence he modified the equation of the photosynthesis. This is the 6CO2 plus 12H2O18. In the presence of light and chlorophyll we get the glucose and 6 atoms of oxygen and 6 atoms of water. Now at last the experiment was experiment by the Robert Hale, the man. Robert Hale in 1937 demonstrated the photolysis of water, which is also the part of light reaction in which the water gets split into the hydrogen and oxygen in the presence of light, and it takes place in the chloroplast. So in this, photolysis of water by isolated chloroplast in the presence of suitable electron acceptors. Electron acceptors are the chemicals which accept the electrons, like ferricyanide. The ferricyanide gets reduced to ferrocyanide, and the reaction is also known as the Hill's reaction. The electron acceptor used under experimental condition is DCPIP. It is, the full form is 2,6-dichlorophenolindophenol. So it is a phenol compound which was used in the Hill's reaction.